through some of them. Plus, I have a list of my questions, and we'll try. The idea is for people to learn about these technologies, and it's really incredible. These amazing collection. So, my first question is for Megan about CRISPR. And Megan, what are some of the ethical implications of CRISPR technology, as well as some of the potential negative impacts of foods edited by CRISPR?
Yeah, that's a great question. And I have a talk that's just about CRISPR ethics. Like, there's a lot. I can move it closer. That's better. Yeah, so there's a lot to say about this, which I didn't really get into. And I think a lot of the ethical discussion has been focused on the human applications of CRISPR so far, which is somewhat for good reason. But I think there are also ethical considerations here in agriculture. And I think, to me, the major ethical considerations are about how we're treating the environment and whether we're being responsible and not promoting monoculture and getting at this, contributing to the lack of genetic diversity that's been brought up previously, and making sure that when companies commercialize products like this, that it's being done responsibly and that people are communicating. I mean, that's part of why I do my job, is I want people to know as much as they can about the science and not withhold or hide information and seed mistrust. Since that was, I think, one of the huge contributors to GM technology getting so loathed generally. So I think there's ethical issues there. In terms of negative impacts of editing in crops, I think you have to think about each individual crop and what you're doing to it. So to me, I don't see a huge risk from the technology itself, which is kind of why I appreciate some of the way the regulation is going so far, is that you should be thinking, in my mind at least, about the changes you're making, not the way you made the changes. And so I think, like that tomato was an example. Like, to me, that's not the most responsible use, because there's not great evidence that that GABA compound is really that helpful, even though it might be appealing to a consumer. But I think you have to think about what the consequences of that might be based on what the actual change is. So to me, there could be some environmental impacts that we need to be thoughtful about. And I know in the US, at least, they're very cautious about like pests and everything in the regulatory sense. So hopefully we don't go down those roads.
Very good, thank you. I thought maybe for this audience that doesn't know much about CRISPR, Megan, maybe you could just briefly say what the sickle cell anemia story is, because it's really amazing.
Sure. So this is an application of CRISPR that has nothing to do with agriculture. There's a mutation that some people carry that causes a disease called sickle cell, where the molecule that carries oxygen in your blood, which is called hemoglobin, gets distorted, and it ends up clumping together and making the cells turn into this sickled shape, or like a half moon. And they're not able to go through blood vessels very well. They get clogged. And so this causes really horrible pain and tissue damage, premature death. It's a really terrible disease. And so it actually is a really good candidate for gene editing, because we've known about the single DNA letter that's mutated in this disease that causes the symptoms for like decades. It's considered the first molecular disease. And so there's been several different clinical trials underway, and there's already results in patients where they've had their blood stem cells edited in the lab using CRISPR and replaced, and they're now basically symptom-free of sickle cell. There are still some occasional flare-ups, since they do still make some sickled cells, but most of the patients that I've heard about, at least, they don't no longer need blood transfusions, they haven't had pain crises, and so it's been super, super promising in humans, in both sickle cell and also in other diseases like beta thalassemia. There's a liver disease, there's eye diseases, there's a handful of clinical trials across the board. So it's a very useful technology, not just in agriculture.
Thank you. Yeah, I have some more questions in here. Let's see. Oh, here, Larry. Have you explored the genetics of disease resistance in soybeans?
No.
And why not?
One sentence. I am truly not a geneticist. I've worked close. We have not explored this. We have looked in collaboration with plant pathologists a few across widely diverse genotypes. And there's a disease in soybean that's called frog-eyed leaf spot. And it's a terribly expensive disease to control with fungicides. There's genetic diversity for it, but it tends to break down because different strains of frog-eye leaf spot develop that are resistant to the fungicides. And so we've looked at a whole panel of different soybean accessions that are vastly different in their genetics. And we have been able to identify a few novel genotypes that apparently have different resistance mechanisms for different strains. But other than that, we haven't looked at disease plant. I haven't. Others may have plant interactions in that way.
Thank you. Very interesting. Hello. So this one, Toru-san, this question is, they're intrigued by your strawberry diversity in Japan and wonder if this is something that could be financially viable way to create next generation indigenous produce in small regions to build niche, high-quality, sub-regional products.
I think that's the idea, because in Japan, each prefecture has their own agriculture scientists owned by the prefecture, and they are eager to make their own brand. So that's why we have so many varieties in Japan for strawberries. And within the prefecture, Tochigi, I mentioned about Tochigi prefecture, but those prefecture is the #1 production of the strawberry. But other prefecture, they are very keen on beating this prefecture. So once it gets very famous, because strawberries are very what is popular to eat, people would like to eat at least once if it's famous. So if it's good, if it's very sweet, or if it's very fancy, people will try to eat. When it's good, they will repeat to eat. And if they want to eat at those prefecture, it will link to what you say, traveling. Yes. So that's the idea in Japan, I think. It may be different from other countries, but that's Japanese.
And could you comment on how did those great variety of strawberry from the different regions, how did those come into being? Those were traditional breeding, I assume. Not CRISPR.
Not CRISPR, no. Well, I'm not a strawberry specialist. I'm a aquaculture man, but I heard that the strawberry in Japan was recently came from Netherlands. Could be. Yes, and yes, in the 20th century. Yes, and well, just like the cherry tree blossoms, they just look for many varieties by breeding it. And that's a really traditional way of breeding, I think. But yes, so what was the question? I'm sorry.
Traditional breeding.
Yes, that's a traditional. Yes. So it's the traditional breeding, and strawberries are cloned fruit because it has what you say they branch yeah. So they replicate, yes. But there are few strawberries that they are from seeds. Some company does that. So for that strawberry, they can use F1 hybrid strategy to keep the priority of the species.
Well, I think it's interesting to tie that strawberry model of Japan. This question is about indigenous products and regional variation. Maybe the person is suggesting maybe each of the different Hawaiian islands could develop a product that's slightly different. You know, we would have and we have some examples of that. The Maui onion is quite famous. And I think there's more, you know, as Richard said earlier today, there's 150 varieties of taro that this was developed by the Hawaiians before, you know, the genetic theories or anything were developed. This is pre-contact. They developed 150 varieties of taro. And so you could imagine, at least I can think that maybe in Hawaii, there could be this kind of regional product, you know, based on taste and quality and the color or whatever. And how about that, Robert? You know, you have the situation in Europe where you have lots of variation in regional products. And how do you get to that? You know, what drives that situation?
Yeah. So I was going to add that we talk about quality and taste, indeed, which is indeed super important. I think it's also nice to highlight, and I'm not exactly answering your question now, I think, but highlight that there's, perhaps you can call it microclimates, basically, so very local, different types of environments, really. And I think creating different breeds suiting for, or genetic lines, whatever you want to call it, suitable for those specific environments is also optimizing the overall production in terms of the growth, performance, but including taste and quality. We see more and more that you cannot just use one specific genetic line for any environment, as I highlighted that also in my presentation. But also maybe, like we say, there's niche products indeed, a specific onion. We have an example where we are breeding a specific Scottish salmon, for example, really local in Scottish waters, performing very well in that area, and eventually sold around the world. But really focus on that area to make sure that you're creating a product that fits. In Europe, yeah, a lot of different climates, you could say. And I think it's exactly the same situation. Yeah, so it's not only about taste, quality, but general performance, I would say. Yeah.
That's very interesting. Selfishly, I'm going to ask a question that I have, and it's sort of bringing together Dr. Purcell's comments about we need to protect the genetic diversity of some of these commercial crops and not just drive them hard. You know, the geneticists want to get to monoclonal situation, the very best, and that's what they're working hard to do. But what that leaves in the dust is the genetic variation in the founding stocks. So I'm almost imagining that in a breeding company, you might have two jobs. And one job is to drive the commercial product as fast as possible to the best commercial performance. But at the same time, you have a second job, and that is to maintain the genetic diversity of the founding stock, because you just don't know what's around the corner in the future for a new pathogen, a new virus pops up. And in your particular commercial stock, there's no resistance. I mean, we saw that in Vaname with Taura, and it just wiped them out. And so, is that an idea that's relevant in the breeding company, or is this just my fantasy?
Absolutely. I think that counts for all breeding companies and not only in animals. The long term is very important for I guess for two reasons. Every time when we improve our animals, and same for plants, it's a step process. So we make maybe. Well, in fish, we can make giant steps. So you could improve certain traits with maybe 10% per generation, or growth, for example. But yeah, it takes a long time to really get any really noticeable improvement in products often. So you need to have a long-term view. But indeed, what we're breeding for today may not be super relevant anymore, or there's new traits that are important that come in. For example, development of a new virus or something that affects your stock. So, and then you need to be able to respond to that 10 years from now, for example, or maybe 20. So it's super important to have a gene pool in your, well, what we call a nucleus, to make sure that there's a lot of diversity left. So you want to, yes, we want to focus on specific traits, but at the same time, we need to maintain a wide diversity to be able to respond to different selection decisions later on. If we would not do that, then the breed or the line would disappear, and we have a responsibility to maintain that stock. So that is important. But so it's indeed a balance. But if I look at, well, at least for fish species, it's maybe we're in an easy, it's a bit easy for us. We have our nucleus, but we can create, with very few parents, we can create many offspring. So for shrimp, it's easy to get, say, 2 million offspring on one female, for example. So you can imagine that I only use very few animals to create I select them on specific traits of interest today, and I use those to produce, on the short term, the offspring to be used this year, basically. But in the meantime, I still have the whole nucleus with all the diversity. So maybe in 5 years later, I would select a few other animals to produce the offspring for that particular year, maybe for a new disease with resistance to a specific new disease, for example. That's still possible. So you can still optimize and at the same time maintain your diversity.
I think you just explained why genetic companies need a lot of income to be able to do what they do, because it's quite expensive to maintain all those stocks, and then the genetic analyses and stuff like that. People complain about, it's too expensive, the breeding stock's too expensive and stuff. But wow, there's tremendous investment to get to that point, right? Yeah.
And everything is about skill. That's true. So the more you can spend on R&D, the better your product will be ultimately. Yeah.
Jim, could I
Yes, sir,
comment
please.
on that? So one of the things that we've run into when we've talked to commercial breeders of soybean that I communicate with is we've talked about introducing crosses that they might normally make to increase the diversity. And Megan touched on this. With traditional breeding, you bring in a lot of traits that you don't want when you make a wild cross or a very diverse cross. And the larger companies are so specialized now. They may have a panel of 500 markers that they use to look at disease tolerance, pubescence color, seed quality, a wide number of traits. And if you go out of that select genotype panel with a diverse cross, that panel no longer works. The molecular markers no longer work. And they're very, very risk adverse to look outside of that because of that. And so they're saying, even if you have something that yields well and has drought tolerance, our company won't use it because it's outside of our panel.
Wow.
Yeah.
A little bit the tail wagging the dog, huh?
Yeah, exactly. Right.
Wow. That's a story, yeah. Okay, I have another audience question. This is very interesting, and we probably all have some thoughts on this. Are there differences in fish health, particularly pathogens, when using fingerlings from captive stock compared to wild-caught fingerlings? And Toru-san, I think you probably know that best. You have some that are domesticated and some that are close to being just wild animals.
Basically no, because if you find the farm disease problems in farm fingerlings, yes, we have the same symptoms with the wild fingerlings right now. But if we go into the breeding to extreme, maybe there'll be a big difference. It's not. We have very shallow or small history about breeding yellowtails, so there is not a big difference between the wild ones. But if we continue breeding, maybe there could be a difference on the traits. So for the growth, we are having a better growth with breeding fingerlings, but at the moment we don't have a big difference. But in the future we might. So we have to keep eye on that, I think.
Yeah, I want to add to that. So I think that's indeed the case, that in time you will start seeing some differentiation, because wild stocks are typically they're produced by natural selection, so they see a lot of different diseases, and animals that don't make it, they will not contribute to the next generation. Indeed, in breeding programs, we have commercial interests, and then indeed growth is, of course, a very big driver. Typically, not always, but you see that animals that grow faster tend to have a little bit lower disease resistance to, not all again, but to some diseases. So there's a negative correlation or unfavorable correlation. And then the trick of a breeding program is to make sure that you're still not only selecting animals that grow fast, but also the ones that still grow reasonably, but also maintain a level of resistance. But ultimately, you are, commercial programs look at what the diseases are, the main diseases are in the today's culture, and that's what you tend to select for more. So yeah, there's a more focused disease resistance, and potentially, indeed, these animals are less suitable to survive in natural conditions. But that's not important if you're talking about culture, yeah. I mean aquaculture, yeah.
Excellent, thank you. Here's an interesting question from the audience. What kind of education do you need to get into agriculture breeding? Do you need a PhD?
Agriculture or aquaculture?
Either. Same. Not really.
For crop science, I think that you can make contributions at a master's level. You probably will not be running your own program, but you would be very much involved in the breeding aspects and variety selection and evaluation, and doing a lot of the genetic selection. But to run your own program, yes, I believe you need to have a PhD.
Megan, would you like to comment on that?
Sure, yeah. I mean, I get asked this a lot by young people studying biology, and I think you definitely need a PhD to advance to a certain point in most companies across all of biological sciences, not just agriculture. There are, rare cases I've heard of where people just get so much hands-on experience that they are valued enough to rise in the ranks without a PhD, but I think it's more of an uphill battle. Depending on what you want, though.
Makes you think, get a PhD.
Or an undergrad degree in that case.
Very good.
That, because if you look at the geneticists around us, I mean, we're working in a team, right? So yes, there are people that have a PhD. Indeed, that's true. Typically for more detailed work in genetics, you could say. But we have a lot of people that do not have a PhD. They have an MSc, and sometimes not even in genetics, but they just grow into the job. But also, if you want to make a breeding program work, it wouldn't work if we would only have geneticists in the team. You need phenotypes. I mean, Larry also showed a lot about characterizing different animals, plants in your case. Yeah, we need people that are able to do phenotyping, so to characterize the organisms, but also reproductive physiologists. If you cannot reproduce the organisms that you're producing for your program, effectively, program is useless. So it's a whole bunch of expertises that we need to make a program effective and implemented. So I would. No, we need everybody, and the very wide IT people think about the databases that you're having. I mean, all those kind of things.
Science is a team sport.
Exactly.
But usually the captain of the team is a PhD. It's not essential to do science to have a PhD, but usually if you aspire to be the captain of the team, you probably need a PhD. Okay, here's really interesting. It seems diversity is the key to resilience, and yet consumers want products that are predictable. Can we have both? Larry, you're laughing the most. You first.
I talked about everything I talked about was about diversity, so I guess the spotlight's on, all right? I don't quite know how to answer that. I want to say that we have diversity in our consumers as well, and that products that they may not. Oh shoot! I'm just bulling. I have no idea. I'll back off.
Very good.
Some diversity to be able to, on the long run, be able to create a predictable product. I had to think a bit about this, but really, if there's, again, in 5 years' time from now, there's a new virus popping up in shrimp, and that's not unlikely, as we all know, or bacterium. They appear every year almost. So we need to be able to respond to maintain productivity of shrimp. And this counts for any species or any organism, I guess. So we need to have the diversity to be able to respond. And in that way, we can ensure that we can produce on the long run.
So it really is a balancing. You have to have the diversity, but then through the breeding, you get the specific product, which is consistent, stable.
Yeah, because you start selecting for this.
So the breeders have to do both. They have to maintain that diversity and get the product. Very good. Okay. This is a good one. Can the panel comment on how new technologies make the entire living world the source of diversity, since a specific protein can be borrowed from any other living source? Can the panel further comment on how protein engineering further expands this base of diversity? Wow. Anybody want to try that? Megan, you know the GMO versus gene editing world. What would you say?
I think they're talking about how now, via sequencing especially, you can start to find the sources of traits from all kinds of crops and species and mine the entire world much more broadly, and potentially apply those traits in commercially bred crops as well. I know I've heard some discussions of indigenous communities talking about traits that they have and not wanting outsiders to sequence, and figure out what genes and variations are causing those traits and then take them and commercialize them and leave the community behind that developed that crop but wasn't sequencing its genome necessarily. So I think there's some interesting conversations to have about who owns the diversity and how to mine it and extract that information and apply it in a fair way. I'm not sure if that got at the question exactly, but that's my take.
Very good, thank you. Okay, Toru-san. Toru-san, can you touch on bluefin tuna aquaculture that you mentioned? Similar to earlier question regarding differentiation for Seriola species, are there observable differences between wild-caught and cultured bluefin tuna? Because tuna are highly migratory with broad ranges. Curious whether there are noticeable differences. So wild type versus domesticated.
Again, in bluefin tuna, like the Kinky University has developed a way to make their own rootstock fingerlings, but most of the farmed bluefin tuna is actually from the wild fingerlings. Sometimes, like in Europe, like Croatia, they catch like 100 kilo or 200 kilo bluefin tuna from the wild and keep it for several months, feeding it so that it will get a good content of fat, which is good for sashimi or sushi. Then they will sell the product. So in that way, there is no big difference between that. But as for the fingerlings produced from the eggs, we still have a difficulty on nutrition. So we see a difference in some traits between the wild fingerlings and the artificial fingerlings. So there are some room for investigation to have proper way of nursery for the bluefin tuna, I think.
Now in bluefin tuna, like either at Nisui or at Kinky University, are there F2, F3, F4 bluefin tuna or not yet?
Kinky University, yes. But Nisui actually quitted artificial fingerlings because it's very hard to handle.
Maybe a return question. Is it a problem that domesticated or cultured species, so let's say tuna or any species, tastes different compared to the wild variety? Is that a problem? Now, the thing is, we want to feed the world, right? So, and we need, because the population is growing, so we need to be more efficient or do clever things to be able that we can feed everybody. So we will need aquaculture more and more, and that means that we will need to be produce our own products. If we eat chicken these days, I mean, no, it doesn't really look like the wild jungle fowl that we started with. And the same for wild boar, or the milk that we're drinking from these black and white Holstein Friesian cows. We accept that, but some way, with fish particularly, we have always this discussion about wild salmon versus cultured. Tuna, we get the same thing. Shrimp, it's not like lobster, no. It's different. Is it a problem? I don't think so. I wonder. So is the question, it's a statement, I guess. But.
I think it's an excellent question. I know in salmon, it's quite actively debated. Oh, I'll only eat wild salmon. Oh, I'll only eat cultured salmon. Years ago, Toru-san told me that, they didn't used to eat salmon sashimi in Japan because the wild salmon, they only had wild salmon, and the wild salmon had a parasite in the meat. So you could not eat raw salmon at the time. Now, in Japan, salmon sashimi is very popular, and it's because it's aquacultured, and aquacultured salmon has no parasites. So there is a benefit of the cultured versus the wild type. That's very interesting to me.
Yeah.
I learned from you.
And can I add comment to that, please? Yes. So salmon is that, and also with yellowfin. Japanese amberjack. I mentioned about the quality. In the spring to summer, the wild caught Japanese amberjack loses weight similarly to the aquaculture because of the spawning. But as we propagate with the artificial fingerling to just go under the size for maturation on the spawning season and can be harvested just after. So the fish will keep the fat content and the meat quality. So that made the market in Japan before, many people thought that farmed fish is not good as the wild type. But now, it has been accepted in Japan. Farmed Japanese amberjacks are more stable and tasty than the wild ones. So it's changing, I think.
I think a small part of that, at least, is that in the farm-raised, our ability to process them, there's plenty of ice and so forth. So the processing maybe is more refined in the farmed product, whereas in the fishery product, maybe, things are out on the open ocean and rocking and rolling, and is there enough ice and all those problems. And so that could be a factor too. But I eat both. I love them both. So, okay. So, oh man, so many good questions, right? Okay. Okay, I did that one already. Oh boy, there's so many CRISPR questions. I don't want to overwhelm us with CRISPR, but Megan's got it. She knows what's happening. Could CRISPR be used to speed up the iteration of phages used as substitute for antibiotics in aquaculture? Great.
So there is work to use CRISPR to engineer phages. I have no idea about their application in aquaculture, but just generally, CRISPR actually came from phages attacking bacteria. It's a big bacterial immune system. And so this is like a big area of research since it has overlap with, I think, with the people that studied native CRISPR systems. I think definitely, you can do phage engineering with CRISPR. There's demonstrated techniques.
Nice. Just FYI, phages are kind of viruses that attack bacteria.
Yes.
And so...
Right, so phages are viruses that specifically evolved to attack and kill bacteria. And so they've been used in some cases. I know a little bit more about like human phage therapy, which is not super widespread, but there have been use cases where someone has an antibiotic-resistant bacterial infection and they can't just use traditional antibiotics for it, so they end up using phages that have evolved to kill that specific strain of bacteria. And there have been a few cases of people that have been like completely saved through this. But it is a really difficult field where that sounds very simple when I say it like that, but it's not super easy to come up with enough of the variation to be able to target every different potentially damaging bacterium and avoid resistance. Like CRISPR is a phage-resistant system, so in that case, CRISPR is working against phage therapy. So it's complicated, but it's an interesting area.
Yes. I might just offer
Thank you, Neil. Excellent. Okay, I have there was a good question here. Yeah, this is for all of us that work with, well, I'm going to say animals, but sorry, Larry. But anyways, what impact will lab-grown seafood have on aquaculture and breeding companies? Lab-grown seafood. Wow. Now there's a big idea. Yeah. You want to try?
I have to, I guess. Yeah, it's very interesting. I think it is one of, well, there is a demand for it as well. We need to recognize that. People often ask, are you afraid of that development? Will it replace animal production? I don't think so. I think it's a very interesting opportunity and a way to diversify animal protein production, if you want to call it like that. But it will be different. And, in the coming, well, I don't know what time span, but in the coming years, it won't replace animal production for sure. It will take a place, and I think that's perfectly fine. But we also know that technically, for scaling up, there's a lot of hurdles to take still to be realistic. And I also personally believe that people still want to eat natural animals. It doesn't sound nice in this way, but I think that's what it is ultimately.
Right. But sometimes in an innovation, you know, we don't think anything's going to happen. Oh, that's just, and then all of a sudden, everything changes. And that's possible with this cultured meat, cultured seafood. I think it's very interesting.
True, and a lot of genomics technology you can still use to optimize and to make more efficient lab-grown meat production, right? So, I mean, in that sense, it's a replacement of the live animal. So, and I think that's perfectly fine.
Yeah, and, you know, from like carbon considerations, grass-fed beef is wonderful, but it's, you know, people are concerned about its carbon impact, and so maybe lab-grown, I don't know. I don't know about that in detail. It's not my field, but very interesting. Okay, here's another question for Megan. Dr. Hochstrasser shared the growth of the gene modification up to CRISPR. What inventions are on the horizon that could be the next phase on this growth spectrum?
Good question. Yeah, the next Nobel Prize winning technology. Yeah, I'm not sure. I mean, I think there definitely can be things that will surpass CRISPR, and they'd probably involve, to me at least, what I would want in a future technology as a kind of genomics thinking person would be molecules that can carry out the entire gene changing process by themselves. So like Cas9 or CRISPR systems, they basically make a cut, and then you're relying on the cell to repair that cut the way you want, and that often doesn't happen, which I didn't really get into that much. So there are some like new CRISPR technologies that can do things kind of like this. Like there's one that makes a specific base edit. Like it'll change a C to a G, for example. But I think the more we can get towards like very precisely controlling the outcomes, the easier things will be to use. But I mean, in agriculture, it seems like having better delivery options, which I think is also true in human applications, would be a huge leap forward. Not necessarily in like the core technology, but in enabling technologies to do what we want them to do in new organisms and in people and different tissues in the body.
Can I ask Megan a follow-up? Yes. So most everything that, as I understand it, you've talked about are qualitative traits. The big things are quantitative traits very often, at least when we talk about from a breeding standpoint. What do you, are there applications for CRISPR for quantitative traits?
Yeah, that's a great question, and I definitely like skim over that for simplicity's sake. But yeah, many traits, like, again, I have more of a human background, like height is controlled by like 700 different places in the genome. And so this is definitely true in food crops and animals as well, where the most interesting traits are very complex. I would say there's been progress made in what we would call multiplexing CRISPR, like making many, many edits at once, but it's still very kind of small and slow. I haven't seen many examples in plants. I know there's a company trying to make humanized pig organs for organ transplants, and they've been able to make, I can't remember the number, but it was like hundreds of edits at once to remove this pig virus that is in pigs that would be damaging in people. I think some of that success is from there being multiple copies of the same virus, so it's like you're targeting the same sequence over and over. But I think we're making progress, but that would be a good next technology to be able to do this in different places at once.
Well, this has been fantastic, and I could go on for hours, but everybody's heard a lot today. And I, first of all, want to say thank you very much to our 4 guest speakers. How about a round of applause?